Again, aloha, my kako. Today have a presentation by Kyhl Austin. Kyhl is a PhD student at UH Manoa in the Rubinoff Lab, and his presentation is titled Rediscoveries and Presumed Extinctions of Hawaiian Moths. So Kyhl, whenever you're ready to start.
Cool. Can you see my screen? It says you are screen sharing, but it's a little slow. Um, yeah, no, I can't see
We
anybody.
we can see your screen right now. It looks like it's not on the PowerPoint.
Oh, there it is. Okay, cool. Is that now in
Yep,
presentation mode?
yep, it is in presentation mode right now.
Cool. Thanks. Yeah, as was mentioned just before this, I'm a PhD student in Dan Rubinoff's lab at UH Manoa, studying native moth diversity and systematics and conservation. A couple disclaimers before I start is that even though this is the HETF webinar, I have yet to actually visit either of the HETF sites on Hawaii Island. I'm hoping to get to Lapuhoihoi sometime soon, but there's been some logistical kind of hiccups along the way, so I haven't yet been able to get over there. Another thing too is I'm going to be referring to moths broadly, but when I say moths, it also includes the two native butterflies. But just because there's so many more moths than butterflies, and saying, butterflies and moths is a bit of a mouthful. When I say moths, just think butterflies too. So let's jump right into it. Quick little background on the history of Lepidoptera studies in Hawaii. So prior to the mid-1800s, there was this impression that a lot of oceanic islands, including Hawaii, didn't really have much of a native insect fauna. And so a lot of European entomologists at the time ignored a lot of Pacific and Southeast Asia in favor of more tropical or perhaps more glorious collecting destinations like the Amazon or Central Africa or Australia. And so for a long time, well after, European contact, Hawaii had been neglected for its insects. And very little was known up until Thomas Blackburn, beginning in the 1870s, who was a missionary to the Hawaiian Islands, began collecting and sending a few specimens back to his home in England for people to look at. And that really caught the eye of a lot of entomologists in England at the time, basically saying, hey, Hawaii has a lot more insect diversity than we realize. We really ought to send somebody to Hawaii to collect so we can catalog and describe this diversity. And so beginning in 1892, R.C.L. Perkins, the kind of preeminent naturalist of the time, was sent to Hawaii to collect insects specifically. He also did a lot with birds. You might be familiar with some of his bird work as well. But he was sent to Hawaii as an exploratory entomologist. And so from 1892 to about 1897, he collected pretty extensively in all the main Hawaiian Islands. And a good majority of what we know now about insects was really based on the material he collected well over a century ago. After he finished that kind of mission with for the English entomologist, he stayed and worked in Hawaii for another about 15 years for the Hawaii Sugar Planters Association, and continued to collect and continue to describe, but not at the same level as in the 1890s. Another big entomologist that specialized in mostly Lepidoptera was Otto Sweezy. For the first half of the 20th century, collected and described a lot of insects in Hawaii, mostly rearing. So a lot of what we know about moth host records nowadays are from Sweezy. And there's this big gap in between basically the death of Sweezy in 1950 until my advisor, Dan Rubinoff, came in the early 2000s, where there weren't many people at all studying Lepidoptera in Hawaii. I should also mention, too, that most of the species that were described from the Hawaiian Islands were not described by Perkins or Sweezy. Most of them were sent back to England to be described by Butler, Lord Walsingham, and Mayrick, who are all English-based entomologists. And then Zimmerman, beginning in 1948, but continuing up to 1978 with the Lepidoptera, kind of synthesized all of Blackburn's, Perkins, and Sweezy's information at the time. So that's kind of where we're picking up now, excuse me. So as you probably know, Hawaii is the extinction capital of the world. There's very few places that can kind of claim such a similar depressing title. But the extinction rates for a lot of groups are fairly well known. So for plants, for example, about 10% of the native flora is extinct or believed to be extinct. Honey creepers, it's about 70%. When you include other forest birds and even some seabirds, it's probably even higher. And land snails kind of take the cake at over 90% in some families. And generally the extinction reasons for a lot of these things is known. So for plants, it's a loss of pollinators, honey creepers, invasive seed predators like rats, and then displacement because of other invasive plants. For honey creepers, obviously it's mosquitoes, to a lesser extent, rats, cats, and mongoose. And then snails, rosy wolf snails, and a few other kind of biological introductions, failed biological introductions have wiped a lot of those out. But for insects, we really have no idea. There's been a few small attempts here and there to kind of quantify what we know or what we think we know about insect extinctions in Hawaii. But essentially it comes down to there's so much diversity and there's so few entomologists studying that diversity that we really don't have a good idea of what's going on. And so for these other three groups that I mentioned, we have a pretty good grasp on what's still around and what's gone. So a lot of you might be familiar with the PEP program, the Plant Extinction Prevention Program in Hawaii, which protects the state's most rare and vulnerable plant populations, plant species with usually 50 or fewer wild individuals left. So for these, we have records of where they're found, how many are left, what islands they're on, what year they were last seen, if they're potentially extinct. The same is true for snails, as there's been a couple of recent papers that have essentially cataloged extinctions in all of the terrestrial snail fauna. And the same is true for birds, right? So you can go through the list and see, hey, 1892 was the last year this species was seen, 1896 for this species. But again, for insects, we really don't know. And part of that is just because of the overwhelming diversity of insects in Hawaii. So this is the most recent Hawaiian terrestrial arthropod checklist, which is about 25 years out of date at this point. You can see that there's over 5,300 endemic species in Hawaii. It's probably closer to about 6,000 now, with a lot of recent work going on describing new species. And virtually all of it's endemic. Upwards of 98, 99% of the species here in Hawaii are endemic. And for Lepidoptera, the endemism is basically 100%. There's about 950 described species. When undescribed species are taken into account, it could easily be twice as high. We're approaching 2,000, 2,500 potential native species of moths in Hawaii. But the only attempt to really catalog a lot of these extinctions or potential extinctions came in the 1980s with Wayne Gagne, who had some funding through the U.S. Fish and Wildlife Service to begin to catalog potential extinctions and reasons for their extinctions in Hawaii. He attempted to investigate 27 of the 950 or so species. And you can see there's a lot of question marks here of things that we don't know why they went extinct or when they were last seen. But again, this is just a small fraction of the diversity. And a lot of this money dried up in the 1980s and not much was done since. But Will Haynes, some of you may know Will, took a few of those because a lot of them were moths in the genus Omiodes that were declared extinct, and investigated a little more closely and actually found 5 of them still around. So even the list that Wayne Gagne came up with in the 1980s was not entirely correct. And so in the years since Wayne Gagne's kind of initial foray into Lepidoptera extinctions in the Hawaiian Islands, the IUCN, International Union for the Conservation of Nature, has kind of taken that and expanded it a little bit, but not a whole lot. So of the 21 IUCN listed extinct species of Lepidoptera, 11 of them are from Hawaii. 22 of the 57 critically endangered species of Lepidoptera are from Hawaii. 3 more are listed as endangered and 9 more are vulnerable. So it really cements Hawaii's place really truly as the extinction capital of the world. But unfortunately, like I said before, that's really just a drop in the bucket when it comes to overall Lepidoptera diversity in Hawaii. So like I said, in those 40 years, the IUCN has kind of treated 45 species total. But if the total diversity as we know it is true, and there's actually 2,000 total species, it's going to take much, much longer to actually fully inform ourselves and categorize a lot of these other species. In fact, it'll take 1,600 years to investigate all 2,000 potential native species in Hawaii. We really don't have the time to do that. And so towards that, I've been working on a database of potential extinctions in Hawaii. But before I kind of dive into what I've been doing, I just want to talk a little bit about Lepidoptera diversity in Hawaii. So some of these you might be familiar with, some of these might not. But insects in Hawaii do some pretty incredible things that they don't do many other places in the world. So there's groups of insects that, there's mute crickets, there's terrestrial damselflies, there's all sorts of crazy things. And the Lepidoptera really exemplify that trend of really unique ecological adaptation super well. So they're snail hunting hyposmacoma, which are these small little micro moths where the caterpillars hunt and tie down native tree snails and eat them from the inside out. They're the only snail-eating caterpillars in the world. There's other groups of hyposmacoma which are fully amphibious. They can spend weeks below water, weeks above water, doesn't matter. There's carnivorous caterpillars. As you can see here, the Eupithecia, which sit and wait for flies or other insects to come by before they reach out and grab them. There's also incredible size discrepancies. So Manduka blackburn-eye, it's the only federally listed endangered moth in Hawaii, is the largest native species, but they also get really small. So you can see Paralopistega up here is a native leaf mining moth, just a few millimeters long. But not only are they ecologically diverse, they're also super just beautiful. I mean, if you look at some of these things, all native, super colorful, ranging from just a few millimeters long to some pretty sizable moths. They also perform some pretty important ecological services. So prior to the introduction of ants and mosquitoes, termites, things like that, insects, especially moths, filled a lot of those roles. So a lot of the decomposition that you take for granted, that's kind of been taken over by termites and ants nowadays, historically would have been done by insects. And a lot of the, or other insects like moths. And a lot of these decomposers would have been caterpillars feeding inside dead wood. So like I said, I've been working towards building a database of Lepidoptera extinctions in Hawaii. And a lot of that has to do with, involves a lot of field work. So here's me. This is a caterpillar in the top left here that I found on Maui in remnant dry forest, but also involves setting traps overnight, collecting moths that way on the right, and also a lot of museum work. So I've been to Bishop Museum, I don't know, probably a dozen times at this point, the British Museum in London, which has a lot of the old Perkins and Merrick material that was sent over in the 1890s, and also a lot of literature reviews. Going through checklists and inventories and published peer-reviewed literature to see what species are reported from different islands and what year they were last seen. So that results in a table like this. So this is a fraction of this total table containing 950 odd species of native Lepidoptera. And then I've color coded it as well, so you can see, the orange is species that haven't been seen in 50 to 100 years. Red is species that haven't been seen in 100 plus years, and then black is species that are still extant or likely extant. And so I've categorized these into basically a presumed extinct for 100 plus possibly extinct for 50 plus, and then likely extant or extant for the species I have records of within the past 50 years. And so just some quick stats when you take the whole database into account. It's not good. So when you include Hyposmochroma, which I mentioned before, the snail eaters or some of the carnivorous caterpillars, there's about 350 species of those. And those can be a little difficult to identify. So I have two different analyses here, one with Hyposmochroma, one without Hyposmochroma. With Hyposmochroma, there are 272 species of the 934 described native species in Hawaii that haven't been seen in 100 plus years. So nearly 30%. And then another 9% of species haven't been seen in 50 plus years. When you exclude Hyposmochroma, you take out those 350 odd species. The numbers go down quite a bit, but it's still kind of alarming. So there's 92 species that haven't been seen in 100 plus years and 58 in 50 plus years. So here is a quick little bar chart. You can see that the extinction rates between genera is not consistent. So these are 34 of the native genera with at least one species presumed to be extinct. Not shown here, 34 genera where all the species are still extant or believed to be extant. So you can see that certain genera, Acropex, Hypena, Paraphasis, and Tritorcleus, are presumably entirely extinct. Paraphasis and Tritorcleus are endemic genera, just one species each. Hypena has 5 native species, all of which are almost definitely extinct at this point. And Acropex, just 2 native species, may or may not be native. I still need to do a little more work to investigate that, but regardless, they haven't been seen in 50 plus years. But on the other end of the spectrum, you have groups like Tamsica, Haliophylla, Eudonia, and Eupathecia, which seem to be doing remarkably well. A lot of these can be found in pretty disturbed forests with very few native plants still around, but in pretty good numbers too. And I'll go into that here in a minute. So, like I mentioned before, some of these are not doing too hot. Hypena, with 5 native species, like I said before, is entirely extinct. There's one species that was introduced as a biocontrol, which can be super, super common. That's Hypena laceratalis. Just to give a picture of what the native species kind of looked like, kind of that body shape. They look different species, but the 5 native species of Hypena are most definitely extinct now. Acrolepia has 4 native species, which are probably extinct, unfortunately. They feed on a really, fortunately rare nowadays native host plant, Iea nothicestrum. There's only 2 of those 4 species that have been collected within the past 50 years, including Acrolepa beardsley, which I found on Maui just about a month ago, which was really encouraging to see. Paraphasis perkinsi, as I mentioned before, just a single endemic species. In really bad shape. This is the only known specimen of that species. We really don't even know what family it belongs to, but until we find more, we really won't know. Endemic to Kauai. And then Spheteris acacia is a weird case of a species that was once super, super common in the 1910s and 1920s. Apparently it was one of the most common native moths. Feeds on the native senna along the coast, but hasn't been seen since 1920. And unfortunately may now be extinct. So there's a couple of reasons why some of these groups appear to be hard hit, or especially hard hit compared to others. A lot of them are really small and difficult to identify. So Batrachirodes is an endemic genus of fern-feeding moths. They make these little cases on the bottom of ferns using the sori. And not only are the cases difficult to spot, but the moths themselves are really tiny and really require careful examination under the microscope to identify them. The same is true for Paralopastiga, which are some of the smallest native moths. They form these really unique, pretty distinctive leaf mines on Alani or Melicope. And each species makes a slightly different shape of mines. This is a Ka'ala species on Oahu that just makes this really messy, unorganized mine. But unfortunately, they take really long to develop as caterpillars. And so it's really difficult to get adult moths out of the leaves because they can take months to finish feeding. And as I said before, Hyposmochroma with 350 plus described species and probably 600 plus total species are really difficult to identify. So we don't have a firm grasp yet on how many of those are still around. A lot of them make these really unique cases you can find on rocks or trees or boulders, even in streams like the aquatic ones. But it's not all bad news. There's a lot of species that can still be found fairly commonly, even in pretty disturbed areas. So in the top left here is a species of Bedelia. It's a native leaf miner. Typically they use grasses, but there's a species that uses Bommeria, native nettle as well. But this is a species we found on Oahu mining guinea grass of all things. There's quite a few species that have managed to switch over to non-native grasses. Which is really encouraging to see. The same is true for the species of Crocodocima, which is the family I specialize in. That the native host is a kuli. It's a native coastal plant, but it's also managed to switch over to the non-native species of a kuli in Sesuvium, which is really encouraging to see. Some of the carnivorous Eupithecia are still really common, even in pretty disturbed habitats, because they can switch to non-native insects. They don't care if a fly is native or not. So here's actually a photo of one attacking a termite. There's no native termites in Hawaii, but they don't seem to and can still be found in pretty good numbers in most habitats. And then these two other genera, Mestalobes and Eudonia, which can be mega abundant even in the most trash disturbed forests. I've been walking through stands of strawberry guava and ironwood and seen Mestalobes flying in the thousands pretty commonly. So most of those seem to be doing pretty good. Both Mestalobes and Eudonia feed as caterpillars in mosses, and they don't seem to care if it's a native moss or non-native moss, which is kind of nice to see. So what's going on? Why are so many species declining? It kind of goes on a case-by-case basis, but they kind of generally fit into one of three categories. The first is introduced predators and parasitoids. So a lot of wasps, ants, things like that really seem to be decimating a lot of native insect populations. For the caterpillars I try to rear out, about a third of them end up being parasitized, and 99% of the time, it's one of three non-native parasitoid wasp species. A lot of moths, too, are specialists on different plants, which are being displaced by invasives like Himalayan ginger and strawberry guava. So species which used to be fairly common on native plants are now becoming rarer and rarer as their host plants are restricted more and more to the most intact forest. And there's a third kind of threat to at least some groups of native moths, which is hybridization. And this is definitely an overlooked threat for a lot of native insects. But in the top right here is a non-native species of Methymna called the armyworm, or Methymna unipunta is the common name. There's actually a small radiation of native Methymna in Hawaii. The bottom right photo is one of those that seem to be becoming rarer and rarer. And they seem to be largely displaced by the non-native Methymna, and it's possible that they may be hybridizing themselves out of existence. The same is true for Kolua, the duck. There's a few other noctuids as well in Hawaii, where there's one non-native species and several native species which seem to be hybridizing. But there's not been a whole lot of work done to investigate that yet, but it is a possible threat. But some good news too. I titled the presentation Rediscoveries of Hawaiian Moths, so I couldn't not show some of my recent rediscoveries. Here's 5 of my favorites, kind of from all over the map. So top left is a species of Pararaptica, which is fairly common on certain parts of Kauai, which had been seen since the 1920s. And I've found it pretty commonly up at Kokee feeding on Kolea as a caterpillar. Top center is a species of Terrastia, which feeds on Willy Willy. And up until I started looking into it, it was actually thought to be an introduced species, which hadn't been seen since the 1920s. But as it turns out, it's a native species that's really restricted to the most intact native dry forest in the Hawaiian Islands. And so it's really good to confirm, that even species that we thought were invasive, or at least not native, are in fact native things and deserve protection. Top right is a species of stem-dwelling micromoth called Meronectria marcella, which hadn't been seen since the 1930s on Oahu. It's actually fairly common in the southern Ko'olau Mountains on Oahu. Carpocynus ceterati's on Oahu, again, not seen in over 100 years. Fairly localized, but can be abundant in certain places given in the right time of year and with the right host plant around. And then Mestalobis semiacrea. I've only ever collected at Pohole Natural Area Reserve, on Oahu, but can be one of the most abundant moths there at the right times of year. But again, seems to be restricted to just that one spot in the northern Ko'olau, or the northern Waianae Mountains. So returning back to the HETF theme, what could be found at Pu'uva'ava'a or Lapahoihoi? These are four of my most wanted species that I think probably still exist in the right habitat, but haven't been seen in over a century. Philodoria pictoriana is a leaf miner on Mamaki, and Sphaerista pernitida, the host plant isn't known, but it's probably Ohe maukra. Probably still exist in certain parts of Lapahoihoi in the right wet forest habitat. And then on the right, Sphaerista flavocincta is a leaf roller on Iliahi, Mapsidius chenopodii, as you may guess, feeds on chenopodium, dry forest species that may still exist at Pu'uva'ava'a. But those are kind of the four possible species I'd like to try to find, but I'm sure there's more, especially in the right habitat on Big Island that can still be found if given enough search time. So that's all I have today. I want to thank everyone in my lab who's helped me out, different people with field work, but also field permits, logistics, access, all sorts of things. People who have helped me with the different museums I visited, the loans, just time spent there, and then a whole bunch of people who have generously let me use their photos for this presentation and others. But yeah, that's all I have. If people have questions, I'm happy to answer them.
That was a great presentation. I did not expect to learn as much as I have, and I'm pleasantly surprised and very excited to kind of hear more about your work. And yeah. Does anyone have any questions? This time you guys can unmute yourselves, you can turn on your cameras, and we can have a little discussion. We have about 30 minutes left of the time block. So if you have questions, drop them in the chat or unmute. I actually have a question. Well, everyone, thanks a little bit more. Would it be easier for your work if the museum specimens were kind of like repatriated back to Hawaii?
Yeah, that's something I've given a lot of thought to. I was in London in October of last year to look at all the specimens that were collected, 120, 130 years ago. And the fact that, I, as a scientist living in Hawaii, have to pay, $2,000 for flights and lodging and food and all that to fly halfway around the world to examine specimens that really ought to be in Hawaii in the first place is a little frustrating. Yes, it would make my life incredibly easy, or easier, I should say, if that material was repatriated.
Okay.
But you've got to be a little diplomatic with how a lot of these things are handled, and so I'm trying to navigate my way through that now.
Okay. It looks like we actually have a couple of questions. And the first one is, how do you bring back critically endangered moths?
It's a good question. It's not something that we have a whole lot of experience with yet in Hawaii. Will Haynes is starting to do a little bit of conservation work with some of the omiotes that I mentioned before, the species that had been examined before in the 1980s. A lot of that involves planting their hosts if their host plants are known. or lab rearing, and then releases. So what Will and some other folks have been doing is releasing species that may be common on Maui and historically occurred on Oahu, but taking the species from Maui and releasing them on Oahu to establish a certain population. So that's what's being done now. But for a lot of these things, we don't really know. We need to figure out their life histories, know their biologies, what plants they need, what habitat they need, in order to conserve them first. And so that's kind of the work I'm doing, is trying to figure out a lot of that basic life history information.
Super interesting. The next question is, or the next comment is, great work, Kyhl. Thank you. Are any native and or endemic moths found at lower elevations, say in our backyards? It's a really good question.
Yeah. So there's what I call the big three: Mestilobes, Eudonia, and Hyposmorcoma, which are the three most diverse genera of Hawaii. But you have species that occur from basically the coast all the way up to the tree line, if not above the tree line on most islands. And there's certain species within those groups that can still be found in, urban Honolulu. Not commonly, but given the right plants and the right habitat, they can still be found. There's a few coastal species too, which can be found fairly commonly given the right habitat.
Great. The next question is, how feasible, realistic would it be to set up a training for field personnel to learn how to ID native moth species? It's a two-part question, so I don't know if you want the next one or just want to answer that one first.
I'll answer the first part first, and maybe the second part after that. It definitely be doable. It's something I really enjoy doing, is getting people excited about native insects, especially moths. As long as people are okay with not knowing species. So a lot of these things are, identifiable to genus maybe, but not species, at least, without really careful, microscope work. And a lot of these things are undescribed. So expecting to say, hey, this is, species, 17 from Hawaii Island, is going to have to be acceptable for some people. But you can get things to family and to genus fairly easily with a little bit of practice. Yeah, it's definitely something I could help with.
Nice. The next question of that two-parter is there a place we could share these observations to support your work?
Yeah, I use iNaturalist for a lot of kind of not a lot of my work, but for some of my work. It's a photo sharing website where people can post photos of plants or insects or really anything, and you have people from around the world who may be experts in that one particular group of plants or animals that go through and then identify whatever species it is that you posted. So I go through iNaturalist periodically for native Hawaiian moth photos, and every now and then I'll find something that I get really excited about, like, whoa, this is a species that hasn't been seen in 100 years. Like, where do you find this? Like, tell me about it, kind of thing. And I've made some good connections, made a couple friends that way, where they've invited me out in the field, like, hey, this is where I found it. Let's go try to find some. Had some pretty good luck that way.
That is super cool. Way to connect to people across the world. So do we have any other questions? We've had some great questions so far.
No question. Just wanted to say thanks, Kyl. That was really cool to hear, and I'm excited to hear about your work on Hawaii Island in the HETF. So thanks for being here.
Thank you.
People are saying mahalo, and then another person said that they would definitely love to take a training. Oh, for those of us who have access to native forests and already upload to iNatural, what series of photos would you recommend taking for group identification?
Oh, good question. So generally, the best photos you can get for moth identification will be one directly from the top, so you can see the wing pattern. A lot of moths rest with their wings kind of held out flat. But another character that I guess you can see pretty clearly in the photo on my screen right here, if I'm still screen sharing, is you see this kind of structure right in front of the eye. It looks like a big kind of bush that's poking out. So having a lateral photo of that structure, which is called the labial palpus, is really useful. The size and the shape and the orientation of that structure, more often than not, can get you at least a family, if not further. So that's so kind of a dorsal photo from above and a lateral photo from the side, if possible, is like the best possible combination. And then obviously, nothing too like blurry and small black dot like what's this moth. High resolution is great.
Sounds like a great opportunity to invest in some new camera equipment. Always taking good photos is always very useful in identification.
Yeah, a lot of these things are pretty small too, so having a nice macro lens really comes in handy. But once you kind of open yourself up to notice small stuff that's flying around you, it's a whole other world of diversity that most people don't notice. It's a lot of fun.
Oh, right. I can only imagine. Do we have any other questions? We have about 20 minutes left. If not, we can close down this webinar. Just want to be respectful of everyone's time. If anything, you guys can always leave your questions in the chat or email us with further questions, and we can reach out to Kyhl and connect you guys with those questions to
Yeah,
get
I
them
also have my
answered.
email here at the front if people want to. I think I had it at this first page.
I think
If
so.
people want to think of questions later and email me, I'm happy to answer those somewhere here.
Somewhere. I remember seeing it. I think it was on your first slide.
There it is.
There we go. So with that, I just want to mahalo Kyhl again. Thank you for coming and giving this presentation to us, and I look forward to you coming out to our sites and seeing what you can identify, and hopefully we can find some that haven't been found in a really long time. Our next webinar is on July 17th. It is titled A Space for Time Substitution Along a Model Hydrological System. And so, yeah, with that, I just want to close it out and say mahalo to everyone.